Yeah, it's my great pleasure to introduce the next speaker, which is Meray Serdar. And Dr. Serdar is working with us in Essen for more than a decade now. And for some reason, she's also getting into touch with stem cells. And her main interest is perinatal brain injury, to be more specific, premature injury. So, Meray, we're looking forward to your talk.
Yeah, okay. Thank you for the kind introduction, and thank you for the opportunity to present the preliminary data of our current study in which we investigate the therapeutic effect of mesenchymal stem cells in our well-established double-hit model of prenatal inflammation and combined with postnatal hyperoxia. I know you are all familiar with that, but I give you a short introduction. Preterm infants represents the largest patient cohort in the pediatrics with a rising tendency, and one key risk factor is maternal inflammation. And preterm infants are exposed to postnatal hyperoxia due to birth procedure, which is potentiated by ventilation processes. Together, these both insults, maternal inflammation and postnatal hyperoxia, increase the risk of an EUP or white matter injury, which is resulting in long-term motor cognitive deficits. Unfortunately, there's no causal therapy to treat a white matter injury, and as the presenters mentioned before, mesenchymal stem cells have shown a neuroprotective and anti-inflammatory properties in various models, and based on this, we investigate the therapeutic effect of mesenchymal stem cells in our double-hit model. Therefore, the prenatal inflammation was induced interperitoneally by a single LPS application at embryonic day 20. 3 days after birth, the pups were exposed to hyperoxia for 48 hours, and at the end of the hyperoxia, the pups received a single dose of mesenchymal stem cells provided by KISI, and at term equivalent age, at P11, the brain samples were collected and analyzed regarding the characteristic hallmarks of white matter injury as depicted here. And to evaluate a long-term behavioral outcome, we performed different behavioral tests at the age of 6 weeks and 4 months. And DTI imaging was also conducted in collaboration with the university in Genf by Johan. And let me now highlight some of our key findings. First, maternal inflammation induces a significant reduction of birth weight, which is exacerbated by postnatal hyperoxia, and this growth impairment persists until P11. And interestingly, we observed a partial recovery in animals in the weight treated with MSCs. And in terms of brain development, the double hit induced a significant reduction in brain volume, affecting especially the deep cortical layer detected by the marker TBR1, which is improved by a single MSC treatment, indicating a neuroprotective effect. We also assessed the inflammation, and the double hit induced a significant upregulation of activated microglia detected by CD68 and EBA1, and an increased EBA1 area, which is enhanced by single MSC treatment. Similarly, astrocyte activation detected by GFAP was also diminished by MSC treatment. While it is still unclear whether these glia responses are pro or anti-inflammatory, we detected a significant upregulation of the pro-inflammatory cytokine EL6 in our double-hit model, which was significantly enhanced by a single MSC treatment. to investigate the specific role of microglia. We isolated CDLB-C positive microglia from our 3 groups and sent the probes to our collaborators in Paris to perform RNA sequencing analysis, and these analyses are in progress. Since microglia are also involved in myelination processes, we also check for the myelination by the marker with the marker MBP, and the double hit model caused a hypomyelination detected by a decreased MBP expression, a reduced fiber length, and fewer intersections, which were improved by MSC treatment. To determine whether this superacute neuroprotective effect of MSCs translated into functional outcome, we also studied the neurobehavior. First, we noted that the double hit induced a delay of eye opening, indicating an impaired neurodevelopment. However, the MSC treatment does not show any improvement. We also tracked the weight development over the 4 months, and all groups show a gain in weight, but the double hit group show a significant reduction in body weight compared to the controls, and we observed again an improvement by MSC treatment. To conclude, the physical developmental milestones are regulated by our double hit model. To assess cognitive functions, we use the Barnes maze to analyze spatial learning and memory. Therefore, the pups were trained for a specific escape hole for 3 days, and at day 4, all holes were closed, and we measured the latency time to find the correct escape hole. And as you can see here, the adolescents and adult rats showed an impaired memory performance, which was significantly improved by MSC treatment. Finally, DTI measurements in the cingulum showed also structural changes. The double hit lead to alterations in the radial density, in the fractional anisotropy, and in the orientation dispersion index, which was linked to altered myelin structure and connectivity. And the MSC treatment improved these parameters, linked to an improvement in white matter structure for a long time. To sum up, our data showed that maternal inflammation combined with postnatal hyperoxia lead to a white matter injury with the characteristic neuroanatomical and behavioral characteristics, which were improved. Many of the parameters were improved by MSC treatment. Additionally, some behavioral tests are ongoing and the analyses are not finished yet. Overall, these findings support the translational potential of MSC therapy for neonatal brain injury in preterm infants. And at the end, I would like to thank my collaborators, Johan, Cindy, and Pierre, and all of the PREMSTEM members and you for your attention, and I'm happy to answer your questions.
Thanks, Meray. Lovely presentation. Other questions? Very clear data. There in the back.
Thank you so much for the presentation. As an outsider to, I guess, the PREMSTEM experiments, I just have a question about the cells, because you use the Chiesi cells in your work. Are the cells thawed and used directly from thawing, or are they put into culture before they're given back in your animal experiments?
Okay, yeah.
And my second question, is the passage number that the cells are used at consistent across all of the studies?
Yeah. Okay, I start with the first question. So before we saw the cells and cultivated them for 48 hours before they were injected in the pups. And the second question was about the passage. So for the whole study, we use the same passage.
This was predefined. One of the very first tasks we had in the whole consortium was predefine certain methods and protocols, and I think this is one of the really cool things that really at I don't know 7 different spots we at least try to optimize all protocols so that everyone is doing the same, using the same cells from the same primary cell bank and the same procedure, and the whole thawing and application procedure was mostly suggested by Chiesi directly, and of course a bit adapted on local environment, so to say, but even cell counting was something we consider. So Paris team and us, we bought a specific cell counter that everyone is trying to do as similar as possible. Yeah, but really good question. Other questions? Then Meray, thanks again, and Meray is also around for the next couple of days.